Now, translation is mRNA to amino acids which make up proteins. And that occurs, all of that occurs in the ribosome. So here is a picture of a protein synthesis. This is a ribosome here. This is a ribosome here. Here's tRNA bringing in my lovely little amino acid, little dot up here. These are my amino acids, and they link up to form a protein. This whole thing would be a protein. This is my messenger RNA that's being read every 3, which is a codon. Don't forget that. Every 3 is a codon, and we code it on here for an amino acid that makes up our lovely protein. Please remember there's 20 amino acids, and the different combinations make up different proteins. There are different types of proteins. Hemoglobin, which carries oxygen in our red blood cells, and then insulin would be a good example too, which is made to regulate our glucose levels, or blood sugar is what we call it. We also can get mutations in this chain. So for example, there are different types of mutagens like cigarettes, UV rays. Of course, UV rays can cause cancer, that kind of thing. But mutations is a change in that sequence or that blueprint of DNA. And it can change the protein. But like I said, 90% of our mutations are either fixed or they're silent, meaning that they do not change amino acids. Because you will notice that on this chart, there are multiple codons that code for the same amino acid. So maybe you might just perhaps get a codon that still codes for the same amino acid and your protein is not changed. There's different types of mutations, point mutation, frame shift, translocation, and chromosomal mutation. You can look at those in your notes and get the definitions of those. We talked about DNA technology. DNA technology is just pretty much using like DNA fingerprints. So here's a DNA fingerprint which is from gel electrophoresis, and finding crime, suspects. Or we also use DNA fingerprint for finding the father of a child, or telling if that is the father of a child. We also have what we call a gene gun, which we can shoot like insulin-producing bacteria. We can take a human gene and put it into those bacteria to make insulin for us, which is wonderful because insulin helps with people that have diabetes and stuff like that.